we're going to be going over chapter 11. Chapter 11 discusses physical and chemical control of microbes. Just for an overview of what Unit 3 is going to be covering, chapter 11 is the physical and chemical control of microbes. So what we're using to control them, like with autoclaving, with wiping down surfaces with Lysol, or washing our hands. Those are all methods of controlling microbes. Chapter 12 is going to be our antimicrobial drugs. So we're going to be going over all the antibiotics, the antifungals, some antivirals, and things like that. Chapter 15 will be our innate immunity chapter. So this is going to be your neutrophils and your macrophages, why we have fevers, why we have inflammation. And then chapter 16 is going to be our acquired immunity, basically what you get with immune memory. And the whole point of vaccines is to generate that. So that's going to be your B cells and your T cells and what their functions are. So it's your lymphocytes. And then whenever we move on to chapter 17, it's going to be disorders of the immune system. So that includes allergies, includes like blood type rejections, the whole reason that we give ROGAM, and autoimmune diseases, things like that. And then chapter 18 is going to be diagnostic techniques that are commonly used to diagnose things. And so that'll be a lot of rehashing old stuff. Chapter 18 is usually like the easy chapter, I'm not gonna lie. But most people don't have a very good background in immunology. Guaranteed that most of you have such a weak background, you probably have no idea what your immune cells really even do and how they function for your cells. Most people don't even know what an antibody is or what it does. So the hope is that after you've taken this class, you'll have a pretty good foundation with that. You'll learn about some of the basics of that stuff in physiology, but it really doesn't focus on it as much. This really is the only place you're going to get to learn about the immune system. So we really want to try to take that home and drive that stuff home. And that way, whenever you are having discussions with family members or friends about vaccines and whether or not you support them or they don't, or whatever it is that you're having discussions with, you'll at least be informed on how the immune system works, which I feel like is something that's greatly lacking in society. Just saying. I got plenty of pedestals. Why did it do that? To stand on in that regard. All right, so microbial control methods. Here are some major terms that are involved relating to microbial control methods: sterilization, disinfection, antisepsis, and contamination. You want to be sure that you understand the difference between these. Right, so sterilization refers to removing all microbial life from a surface or a solution from something, okay? So even endospores will be destroyed. Endospores are extremely resistant. In fact, they're the most resistant of all of the aspects of microbes. And they're meant to be. That's what the microbes make them for, is to be resistant and hardy and everything like that. And so they can resist a whole lot of things, a lot of which we're going to learn about today. But in order to sterilize something, to completely remove all microbes, you have to get rid of even the endospores. And so sterilization, complete destruction of all microbial life. What you'll notice is that if you go buy things like Lysol wipes or like bathroom sprays or things like that, it'll say 99.9% effective. They are not called sterilizers, they're called disinfectants because they don't get rid of endospores typically. So they're only 99.9% effective. They're typically going to leave behind a lot of the endospores. So that's disinfection. So disinfection is destroying most of the microbial life. Generally, the thing that's not destroyed is your endospores. Viruses are pretty easy to destroy. So don't ever think that like viruses, because they're so tiny and they don't need to have parts and all this stuff, that makes them more resistant. That's not true at all. Generally speaking, bacteria are more resistant than viruses, but endospores are like a whole next level. They're really resistant. So disinfection, we're eliminating most of the microbes on an inanimate surface. Typically, disinfection is for inanimate surfaces. If you're talking about live living tissues, living surfaces, that's antisepsis. You guys know this word. You're probably more familiar with it than you think, like an antiseptic. So this would include like applying alcohol to someone's skin before you do a blood draw, or starting an IV, using chlorhexidine or something like that, iodine, preparations before surgery. All of that stuff is antisepsis. Okay, so it's the same thing as disinfection, but it is just for living surfaces. Then we have decontamination. So the difference of decontamination from disinfection. Disinfection, we're destroying the microbial life. Decontamination, we're physically removing it. So this could be by filtration, that would decontaminate, or when you wash your hands. The soap isn't typically, it could have some aspect that could kill some microbes, but its real major purpose is to just physically remove them. And so that's a decontamination process. And decontamination is something that can apply. We can also call it sanitization. Hand sanitizers, things like this, often are usually associated with helping physically remove the microbes, and it could have aspects of the antisepsis as well. Okay, so decontamination, mechanical, like physical removal of the microbes. Disinfection, you're destroying the microbes. All right, so here's a chart, but this is an incredibly useful chart for your studying for this chapter. You better guarantee that your exam questions are going to ask things like, would this method sterilize? Would this method just disinfect? You know, things like that. So you'll have multiple choice, of course, but like you're going to need to know whether certain methods are able to sterilize or not. And so this is our chart that's going to break all of that down. Our physical agents and our chemical agents. I typically lump in the mechanical with the physical, just saying. So heat, radiation, and filtration are our major groups of our physical agents for microbial control. There's other ones that are on here, like using cold or desiccation, or using salt, which would be osmotic pressure, things like that that are used for preserving things. That isn't on this chart that you still wouldn't be aware of, but this is a great breakdown of like the major points of that. Moving on, again, just to reiterate, bacterial endospores are considered the most resistant microbial entities. And that's usually the goal of sterilization, is to destroy these guys. If you are able to destroy endospores, you're going to be destroying everything else too. So if you're capable of destroying an endospore, you're almost certainly destroying everything else. So if you're destroying other things but not the endospore, that's the disinfection, the antisepsis stuff. So sterilization really is referring to like completely removing or destroying all living organisms in it, including the endospores. So any material that has been put through that process of removing all living microbes or destroying all microbes, that thing would be called sterile. And sterilized products that we would need would be like surgical instruments. We can't be introducing microbes from our environment into the body where they don't belong. And we often have tissues in our body, like blood is one of your tissues. So your blood should be completely sterile. It shouldn't have any microbes. There's no microbiome in your blood. So if we are introducing microbes into that, you could consider your blood as a whole mess of just open stadium seats. Just microbes just waiting to take over that. And so you can't be introducing microbes into that environment. So that's why when we talk about surgery, why it has to be as sterile as possible. Disinfection often is just not enough for surgical type of settings. So things like surgical instruments and syringes, that we would absolutely want this to be sterile. And there are some foods that we would want to have some sterility as well. That could be like powdered foods and things like that we want to be sure that microbes didn't get dried along with it, that sort of thing. All right, disinfection, again, removing as many of the microbes as possible. So disinfectants are usually used on inanimate objects. And so if you are cleaning your tabletop and you're getting ready to make food, using Lysol on that and getting rid of 99.9% of germs is fine. Being around people that have an otherwise healthy immune system, cleaning with a disinfectant on surfaces is perfectly acceptable. Generally speaking, what's left over isn't going to have a chance to really cause infection or anything, as long as you're not introducing it into the tissues of the body, right? So disinfection is perfectly fine because sterilization is actually pretty difficult to achieve. And the processes that are involved in achieving sterilization can often lead to ruining, living tissue, obviously, because if it destroys endospore, it's probably going to destroy your tissue also, or destroying the equipment or the item that you're trying to sterilize. So some things will just be disinfected. It just depends on what you're using it for. Antisepsis, again, this is like disinfection, but for animate surfaces. So what's sepsis? Sepsis is the growth of microbes in any of the tissues. And then asepsis is basically any practice that prevents contamination. So I refer to it as any process that prevents, asepsis is any process that prevents contamination. We are not just talking about only introducing true pathogens here. We're talking about any potential pathogen. Any microbe that doesn't belong in an area could potentially become an opportunistic pathogen, even though it's not a true pathogen all the time. Remember, true pathogens always, they cause disease, even in healthy people. And that's what they do. So aseptic technique, we try to keep things as sterile as possible. We can't always maintain pure sterility, but the whole idea of aseptic technique is to prevent contamination. And an antiseptic or antisepsis. We're referring to chemicals that are used on body surfaces. Again, using alcohol to prep, before you do a needle stick, an iodine, chlorhexidine, all these things are antiseptics. All right, cidal versus static. We went over this with our Kirby Bauer, so we're already aware of what these suffixes mean. But cidal agents are things that kill the microbes. Static agents, those guys are the guys that could just inhibit the growth, right? So germicidal, bactericidal, fungicidal, virucidal, sporicidal, these tell you what they're going to be killing. So you can be really general and say germicidal or microbicidal. So you're talking about like just microbes in general. If it's sporicidal, that means that it will destroy endospores. So static, it's the same concept. It's a suffix that we'd add on to the bacteria or the fungi or whatever. But this basically inhibits the growth of the organisms, but it doesn't kill them. Decontamination, which we can also call sanitization. This is anything where we have actual removal, again, of the microbes, so physical removal of the microbes or mechanical removal, however you want to think of it. But we're literally removing them physically from that environment. So washing your hands removes them physically from your environment. This also often is used to talk about like removing debris as well. So if you go outside and you are walking on the grass without any shoes on and you get inside and you want to wash your feet off because you got mud on your feet, you're removing the microbes, but you're also removing like the dirt and things like that. So decontamination can refer to that as well. So removing the debris that's associated with it. So there's different levels of our medical devices for microbial control. We have critical medical devices, semi-critical, and then non-critical, and they're basically defined by how sterile they need or how disinfected they need to be used. So we consider critical medical devices anything that would come into contact with sterile tissues. So we're talking about scalpels and things like this. So anything that has to be sterile. We can't be introducing any microbes. You would want like your needles for venipuncture, you'd want them to be sterile. That's a critical medical device because we're inserting into a sterile bloodstream. So that's an example of that. Semi-critical medical devices. These are things that will come into contact with your mucosal membranes. So these are more susceptible to microbial transmission. So either transferring it to the patient or from the patient to the environment as well, due to the moisture in these areas. So semi-critical is kind of in the middle because of the mucosal membranes. So this would be things like respiratory equipment, things like that. And then non-critical medical devices, things that will come into contact either not with the patient or with their intact skin. So this could be things like door handles. It could be straps that are used in situations. It could be, what are they called? Like leads and things like that, that would be used typically in intact skin areas, but could have come into contact with the patient. So how do our chemical agents work? Our chemical agents or physical agents, both of them, physical or chemical, as well as our antibiotics, they're going to target these four things. The antibiotics just have a fifth category, and we'll talk about that when we get to the next chapter. But it's either going to target the cell wall, the cell membrane, which is called the cytoplasmic, or the plasma membrane, or the cellular membrane, however you want to call it. And then cellular or synthetic processes. This could be making DNA or RNA, as well as making proteins or the actual proteins that are already made, denaturing them and things like that. All right, so let's talk about the cell wall first. We can cause damage to the existing cell wall. We could block the wall from being synthesized. We could digest it completely, or we could just cause partial breakdown of the cell wall. All of this stuff will lead to lessening of the integrity of the bacterial target cell, right? So anything that has a damaged cell wall will be more susceptible to the other chemicals that we could be. Usually we use it in conjunction with other things. So examples of things that can affect the cell wall will be detergents and alcohols. Things that affect the cell membrane. We know that our membrane is selectively permeable on purpose, so the water should be able to pass quite easily and freely through the cell membrane. And we learned about there were other very small non-polar molecules that could pass as well, but that's controlled for a reason. And so if you can disrupt the integrity of the cell membrane, you will lose that selective permeability, and then things can flow freely in and out of the cell, and we can't control what's going in and out of the cell as well. This itself could potentially be deadly, but typically, again, this is like opening up the cell, and the best method after that would be also introducing another thing to it that you open up a cell and now this chemical can get in and do its action. So it's usually, again, going to be combined with something else. Surfactants are polar molecules with hydrophilic and hydrophobic regions. This should sound familiar. So if we had a hydrophilic, a water-loving head, right? And we had the hydrophobic tails. Does that look familiar to anybody? Looks sort of like a phospholipid, right? That's the idea, is that the tails here would not be water-loving. They would be water-fearing. So this is definitely a heart where I'm going to draw a line through it so we can pretend. It looks like an apple, but pretend like it was a heart. So these are just like your phospholipids. And how they're going to work is by, well, I always forget that when I do this, I have to click a million times to get out of it. So our surfactant molecules, again, we have them shaped a little bit differently because they're made up of a different chemical compound, but they have a similar aspect to our phospholipids, which allows them to sneak in between. But it disrupts the ability of the lipid bilayer to stay together when they do that. And so the lipid bilayer will basically fall apart and allow things to move into or out of the cell, which is what's depicted down on the bottom down here. All right, obviously, if you're trying to multiply your cells, you're going to need to be able to make DNA and RNA and protein, because every time you make a copy of your cell, you're going to need to rebuild all the parts. So if you can inhibit any of the processes in that, so inhibiting DNA replication or inhibiting transcription or translation, that those are processes that some of these chemicals or physical treatments can interfere with. So we've talked previously about the importance of the protein's shape, and that a lot of our protein is kind of held together by hydrogen bonds and then attractions and interactions between those R groups on our amino acids. So the native state of that protein, again, is that functional state, and denaturing means that you have changed that functional state somehow. You can do this by adding heat that would disrupt those weak attractions. You could also add solvents. You can change the pH. All of these things can affect the stability of the protein, its shape, and therefore its function. We can also introduce metallic ions, and the metallic ions can basically squeeze into the active site and basically act as like a competitive inhibitor, so block the substrates from getting in there. So here's some images of that. So here's our native normal state here on the left. We could have complete denaturation, so the complete structure is broken down. We can't interact with our substrate anymore. We could cause it to have a different shape just by adjusting pH and causing our interactions to basically be disrupted in a different way. Or we could maintain the shape, but just block our ability to bind with our active sites. These are all ways that physical and chemical methods could potentially disrupt our protein function. Let's get into our physical control methods. Heat is obviously the most widely used one. It's actually the reason for like making sure that your food is cooked thoroughly and things like that. So heat is the most widely used. We also have radiation, filtration, possibility of ultrasonic waves, which we won't really talk about in this chapter, and then cold. Osmotic pressure is another one. Starting with heat. So first is moist heat. With moist heat, we're adding in water during the heating process. Typically, moist heat treatments will range from 60 degrees Celsius up to 135 degrees Celsius. Keep in mind that room temperature is going to be 20 degrees Celsius, body temperature is 37 degrees Celsius. So we're talking about temperatures that are approaching and surrounding like boiling, which is 100 degrees Celsius. So around there. We can add pressure to help rise the temperature. We add steam for the moisture and that sort of stuff. So the moisture is actually really important. The reason the moisture is important is for helping the proteins coagulate, which disrupt, so they'll stick together, which will disrupt their ability to function properly. So adding in the moisture really helps. When we use dry heat, the low moisture content, the moisture was helping with destabilizing the proteins. So if you take the moisture away, you're gonna need higher heat. So here we're talking about, so if moist heat, we needed 60 to 135. Dry heat, you're gonna need 160 and up pretty much. And definitely up if you're trying to sterilize. So in this situation, we are dehydrating the cell, and potentially incineration can result as well, which we know we do that with our loops in the lab with our Bunsen burners. Incineration is oxidizing all of those organic molecules and turning them to ash. So you produce carbon dioxide, and then the little ash is what's left over from that process. We compare moist and dry heat and their effectiveness. For moist heat, we have 121 degrees Celsius for 15 minutes is typically very commonly used to sterilize things. So most autoclaves will operate at 121 degrees Celsius with pressure at 15 PSI. So it's more pressure than what our normal environment is. And then the time to sterilize, they usually put it in for 15 to 20 minutes. 20 minutes is the typical norm. And so you're just having to expose something to moist heat and pressure, and then you have it sterilized in 20 minutes. So, but if you're wanting to use dry heat, yes, there it is. If you want to use dry heat at the same temperature, 121, you'd have to go for 600 minutes. So it's 10 hours. So it's quite the difference, right? So that puts into perspective how much more effective it is to add in the moisture with our sterilization. Obviously, not everything can withstand the moisture, though. Certain metal instruments might be likely to rust, so it would interfere with the integrity of those things. The problem with heat, moist heat especially, because you need the moisture to get to the surfaces in there. So the problem with moist heat can be getting inside of an apparatus with the moisture for it to be effective. And so dry heat can be used on things that can't be exposed to the moisture, or that you can't get the liquid, the moisture inside, but at least you can expose it to the heat, the temperature, and it should still be effective, but you just have to treat it for longer. And then we have other methods for things that just can't be treated with heat at all. Obviously, some plastics or things like that would melt. Paper might burn, so there's stuff like that. Right, so bacterial endospores, because they are so resistant, it requires temperatures above boiling. So I want to make this very clear. Boiling does not sterilize, period. All right. There are ways you can make boiling sterilize things, but it would involve understanding the cycle of how endospore organisms grow. So you'd have to like boil it and let it sit for this exact amount of time, and then boil it again when the endospores are coming back out and thinking it's safe to come back out again. And you have to do that several times in order to effectively sterilize. It can be achieved. It's a process called tyndallization, and it's not something that we need to know for this class. But generally speaking, boiling something for 30 minutes is not sterilizing it, it's disinfecting it. For most situations, that can be acceptable. So we're gonna go on. So it's really hard actually to get rid of the endospores, but the vegetative forms, the multiplying forms of these bacteria, the active ones, they're going to be more susceptible, but then it depends on the species and like its actual makeup and everything like that. It's not like you just say endospores require this and then vegetative cells require this. It just depends on the cell and the species and everything like that. So, all right. So this brings up the concept of, if you're wanting to understand how effective your heat is at getting rid of your microbes, thermal death time and thermal death point. So these are basically an assessment of how much time it takes to kill a microbe versus at what temperature it takes to kill a microbe. So the thermal death time is the time that it takes to kill a microbe at this temperature. So it's a temperature that you are looking at specifically. How much time does it take to kill the microbe at that temperature? And then the thermal death point is the lowest temperature that you would need in order to kill a microbe in 10 minutes. So whatever your microbe is, what's the lowest temperature that you can expose that microbe to for 10 minutes to ensure that it is killed? All right, so this is different for different organisms, but that's what these terms are referring to. Let me mute this. All right. Again, boiling is for disinfection, not for sterilization. So a single process of boiling, even for 30 minutes, is not going to sterilize, but it will kill most pathogens. So most non-endospore organisms will be killed if you boil it for 30 minutes. It gets pretty close. It gets you to that 99.9%. This is still useful, obviously, if you have somebody in your household that is sick, or if you're trying to treat a wound or something like that, then boiling can be very close to as effective as you would need it in a day-to-day basis. And it's definitely better than not cleaning something. So there are advantages and there are disadvantages to boiling. Pasteurization is another way of disinfecting. So if you've ever drank milk or most other liquids that come pre-bottled, then most of them have been pasteurized. And so what pasteurization is cycling from a warm temperature to a cool temperature repeatedly. And you do it several times over and over and over again. I had a student in my morning class yesterday who asked me why Brahms milk tastes better than other milk brands. Anybody in here drink lactate or lactose-free milk? Okay. So, I don't know. I hate milk, you guys. I hate it.
don't like milk.
Yeah, so I don't like it, but I'll tell you this. Lactate tastes way better to me than any other milk. And it's not because it doesn't have lactose in it. Because to me, it still tastes like milk should taste. If you ever drank a lactose-free milk, it tastes the exact same as milk, but it just doesn't have that funkiness to it. Why is that? Because they ultra pasteurize it. It has less microbes in it. And you know what else? Brahms ultra pasteurizes their milk. It's not lactose free, but it's ultra pasteurized. That's why it tastes better. That's why Brahms milk tastes better than Highland or anything like that. Yes. Mhm. Yeah, you can taste the microbes there. I don't know, man. Yeah. Yeah. What? Uh-huh. Yeah. Mhm. Yeah, you can tell the difference. Is this milk okay? You can taste the difference. And so if I would recommend that if you guys are milk drinkers at your home, buy Brahms milk one time and then come and tell me that it doesn't taste better. I bet you it does. It's because they ultra pasteurize it. So what's ultra pasteurization? It's doing it more or doing it at higher temperatures. So their pasteurization process exceeds the recommendations from like USDA and FDA. So Brahms exceeds those standards. And so that's what ultra pasteurization is to exceed those standards. If you buy lactate milk, y'all, it's still milk. It's just they have added an enzyme to break down the lactose in it. And so to me, it tastes the exact same, but like cleaner is what it tastes like to me. And so it doesn't have that off-putting taste that regular milk has. I don't drink a lot of milk. I buy it for like my Oreos or my coffee. That's really the only time I have milk. Yeah. Yeah, or cook with it, right? I make my macaroni and cheese with milk, but like I don't drink it. My ex-husband's family was Catholic, and I mean, they had 7 kids in the family. And so they would buy like gallons of milk and freeze them. And like they drank milk with every meal. And that's what they drank. Like whenever you had dinner, they would have like a gallon of milk out for people to drink. And I can't do it. Like I would be like, I'm good with water. Yeah, that's me. Mhm. Right. Yeah. So a lot of people have that problem too. Is like you buy milk because you need it for like macaroni and cheese or something or cereal or something like that. And you just don't drink it all. Buy lactate. Okay. Because it will last months after you open it. I'm not kidding. So if you haven't bought lactose-free milk or ultra pasteurized milk, buy it. If you've been not using your milk, buy ultra pasteurized milk. It will last months. I'm not kidding. Like the sell by date, the last time, like we went and bought like lactate milk, like in early February, and my sell by date or use by date is like March 25th, and it won't go bad until then, even after you've opened it. Like, but regular milk, you don't even have to open it. Yeah, it's gonna go bad within a couple weeks. What was you? Did you have something to say?
I noticed that when I bought milk, I bought regular milk and with that like a week, and I bought Fairlife milk.
Fairlife ultra pasteurizes as well, from what I understand. Yeah. So you can look that stuff up. That information is available, and you can also look. It should say on the label whether it's just pasteurized or whether it's ultra pasteurized. And so if you care about that stuff, I'm not saying you have to, right? But if you're somebody that doesn't want to waste your milk and you have to buy some, buy ultra pasteurized. It'll last a lot longer. You can tell. Yeah. Yeah, you can tell. You can tell. So I've been buying the lactate milk for so long. My husband is lactose intolerant, so that's why I bought it in the first place. And it changed my whole view on milk because I hated it before. But now that I have the lactate milk, it tastes better, it smells better, and it lasts forever. And like we won't use it for so long. And I'll like pour it in a cup and like, yeah, this can't be safe. I'll smell it, I'll taste it. It is fine. It tastes better even than when you buy a fresh pasteurized milk. People will try to tout about saying that raw milk is so good because it'll be good for your immune system or your gut biome or whatever. Good luck with that. Gross is what I have to say about it. There's so many other microbes that aren't being filtered out or pasteurized out, things that could be potentially dangerous, that are actual pathogens. The whole reason for pasteurization is to remove the pathogens, right? We're not removing all the microbes. We already said it's a disinfection process. But the whole idea is to remove the pathogens and get it to what they consider an acceptable level of microbes in your milk, per FDA and USDA regulations. So it's just cycling between warm temperatures and cool temperatures. You never get up to boiling because they don't want to destroy the integrity of the proteins in the milk. So that's the whole reason for not going up to boiling. We don't want to denature the proteins of the milk. We want to retain that. But we do want to get rid of the microbes if we can. So that's cycling between cold and hot helps with that. Kombucha, for example, we know kombucha has microbes in it that are good for your gut, but it's been pasteurized, I can assure you. All right, so we have steam. So this is moist heat method, steam under pressure. So we did moist heat with boiling, we did moist heat with pasteurization, and now we're adding in pressure. That'll let us get the temperature above boiling. Otherwise, it's pretty hard to get water to boil above boiling temperature. It's just going to boil and then evaporate. But in order to force water to get above boiling temperature, you can add pressure with that. So it's a property that we learn about in our chemistry classes. But the autoclave is an instrument that allows us to achieve that. So it's basically a metal box that holds in the pressure, and then you pipe in steam, and that helps raise the temperature. So 15 psi at 121 degrees Celsius for 20 minutes is commonly used for glassware and things that are going to be heat resistant that can be exposed to the moisture. So this is what a typical larger autoclave would look like. You can have desktop autoclaves that are commonly used in like dentist offices and things like that. When I worked at, volunteered for like the Humane Society, I volunteered at their spay and neuter clinic. And I mean, they crank out some surgeries at that place, y'all. I mean, it's back to back. Like the surgeons just stand there for 8 hours a day constantly. They never leave. Like they just bring in new animals and just cycle them through. And so it's like crazy because they'll just like had a bucket on the floor that they would just drop their instruments in. And then I would come into the room and pick up the bucket every now and then clean off the instruments and then put them in the autoclave to be autoclaved and then bring them back to them in packets once they had cooled. So, and I mean, they would just cycle through it constantly. And so you can reuse your instruments like scalpels and things like that. Potentially, if you're autoclaving them, there are, of course, items that we have that are single use, like our syringes, things like that aren't going to be autoclaved typically. So those things we would want to have to be single use, and then you would dispose of them in your sharps containers, things like that. We'll talk about how those get sterilized in a moment, but it's not by autoclave typically. But autoclaves are very commonly used in medical environments. They are like your number one go-to for your reusable instruments. You can even autoclave like cloth. So if you have moist cloth, you can autoclave cloth and it will sterilize the cloth. All right, dry heat methods. This would include incineration, like what we do in our lab. Bunsen burners get up to like 1,870 degrees Celsius. So now we're talking like that's some heat. Most incinerators and furnaces operate between 800 to 6,500. The bigger the item that you're trying to incinerate, for example, if you're trying to incinerate a body for cremation, then you would be using it closer to that higher end of that to get everything ignited and then turning that organic matter into gas and ash, and through a process called oxidation, just powered entirely by heat. You can use dry air in hot air ovens. So this includes like your oven at home. So you can actually sterilize things at home as long as it's heat safe by running it at like 350 degrees Fahrenheit for like 4 hours. And that could actually sterilize things at home. So you'd want to be sure that it is, contained and closed that whole time. And the second that you take it out of the oven, if it's not packaged in something, you're exposing it back to the environment and it becomes contaminated immediately after that. So you have to have ways to store it if you were to sterilize, but just in case there's some sort of emergency situation where you might want to have something sterilized, you're going to have to wait the 4 hours, but you can still sterilize it at home if you needed to. Okay. But we use this for things that are sensitive to the moisture of the autoclave. Cold. So cold is not useful for sterilizing things. Most things aren't going to die from being frozen. And I mean most. So even when you buy your chicken at the store, if you go and freeze it, most microbes are going to survive that. And so that doesn't mean you're eliminating anything by freezing it and thawing it back out again. Some things like helminths, like our parasites and things like that, those don't survive being frozen very well. So if it's been frozen and thawed out again, usually our helminths don't survive that very well, but microbes can. So just keep that in mind. But it does stop things from growing and multiplying. So that's why if you freeze something, it doesn't continue to go bad in the freezer, right? You've stopped it from growing. That's what cold is useful for. There are microbes that flourish at freezing temperatures still, and they're called psychrophiles. So obviously you can't, control. I know the word is crazy. It's a crazy word, but the psychrophiles means that they love cold temperatures. I don't know where psychro comes from, but yeah, apparently that's what it means. All right, desiccation, drying things out. We know this with like beef jerky. Beef jerky is usually also a little bit salty as well, but the drying out process is useful for this. You can actually combine freezing and drying in order to make some powders. And that's a process called lyophilization, which you may have heard of, but there's sometimes you'll get these vials that have powder in them, and you'll have to inject saline or something into it to mix up the powder so that you can use it. That powder has been lyophilized. So they dry it out in a vacuum at freezing temperatures, and that helps preserve whatever it is. All of our samples that we use in our microbiology lab came from lyophilization. So I buy vials of powder, and then I just reconstitute them in broth. Sometimes they grow and sometimes they don't, but sometimes it really literally depends on like the weather outside, which is, I saw this meme on Instagram where it was like microbiologists looking at bacteria, and it's like bacteria in the wild grow on everything. They could get anywhere. They can't be stopped. They're, defying the laws of life and everything like that. But you try to grow them in the lab, man, they are so picky in the lab. The temperature, like the exact concentration of your media, all these things. Like I have so many problems with them. It's ridiculous. All right, so radiation, moving on. Radiation is a physical control method as well. So here we are bombarding the microbes. Well, if you're trying to kill microbes, right? You're bombarding the cells with basically excited electrons. And so you're putting energy into the atoms, and it causes the atoms to release electrons. And these electrons will fly off and then cause damage to the proteins, the DNA, the RNA, every membranes, every molecule in the cell can be destroyed by radiation. This is especially true of ionic radiation, which is what I'm describing. When you're exciting the electrons and they start to shoot off because they're so excited because they're being moved into higher orbitals, and then they get eventually, with enough excitement and energy, they'll fly out. So this is what we get with X-rays and gamma radiation. So that is ionic radiation. UV radiation is non-ionizing radiation. There are strong enough ionizing radiations that can pass through any material. So here we're talking about like the radiation given off by like the elephant foot in Chernobyl. If you know anything about Chernobyl, how there's a nuclear meltdown. And then because that reactor had a meltdown, it basically left like this radioactive, they call it the elephant foot. So it's what's left over of the radioactive material. And it is so radioactive. It's going to be radioactive for like forever, basically. But it's slowly decaying. And as it decays, it releases off this radioactive energy. If you were to go near it, what's actually happening is that energy will cross through your cells and all of the molecules that are in your cells, your membranes, your DNA, your proteins, and it's going to damage all of those molecules as that energy hits them and literally break them apart. This can be so damaging that your body cannot recover anymore, because you'll damage your body's ability to heal as well. So when you have people that are exposed to radiation, they can like go up and hang around it for a little bit and then walk off. And a couple days later, their body, all the cells were so damaged, they're starting to die, and your body just has no ability to heal it whatsoever. And the DNA is damaged and everything like that. So you're lucky to survive situations like that. And of course, you could have damage that will be caused to the DNA, that if it isn't repaired by your body in time, can lead to cancers that can develop as a result of exposure to radiation as well. Yes. Yes. Yeah. So X-rays count as ionizing radiation. X-rays are weak enough that you can block them with lead. It's one of the barriers. It's so dense that the X-rays can't pass through it. But gamma radiation, like what we have, like the high level gamma radiation, like what you have at like Chernobyl and other nuclear reactors and places like that, can't be blocked by anything. You could stand behind 5 plates of lead and it's still going to get through. So it just depends on what basically wavelength you're dealing with. So X-rays can be blocked. So that's why if you're working in a healthcare field, most people can withstand a little bit of exposure to the X-ray radiation and survive it just fine. As long as you're only getting an X-ray like, once or twice a year. But if you're a healthcare worker, you might be giving X-rays or be around X-ray machines pretty often, especially if you're a radiologist, right? And you're the one operating these machines all the time. That's your job. So that's why we have to have special walls or special like aprons and things like that that you would wear to protect your body, especially your sexual reproductive organs, because those cells are going to be the most at risk for damage from that radiation. So yeah. So that is ionizing radiation. Yes.
What about like airports or like airplane?
Right. So there's going to be radiation anytime that you're like going through those screens at the airport. If you're flying really, really often, I mean, like constantly, then that could potentially be a risk. But the X-ray machines that they use at the airport are super low level radiation on purpose for that reason. So I mean, if you were literally flying twice a day, every day, then I could see it being a problem. But they've done, research to make sure that most people that even people that are flying often for business, like they're not usually flying twice a day, every day. So when you talk about like pilots and stuff like that, they don't go through those security checkpoints. I don't know if you didn't notice, but they don't. So they are going to be, exempt from that sort of thing. So they're not getting exposed as often. And obviously those things don't emit enough radiation for the people that are working at those stations to be exposed. It's such a low level and it's contained within that area that for them, it's just not a problem to work around. But yeah. That makes sense? So moving on to non-ionizing radiation, this would be like UV radiation. UV radiation still excites your molecules, but what happens with UV radiation, they're not shooting off electrons like we have with ionizing. With UV radiation, which is non-ionizing, so when we shoot off those electrons, it creates ions, right? Remember, an ion is something that has gained or lost an electron and gives it a charge. Well, if you gain or lose an electron just by shooting it off from energy, it creates ions. But non-ionizing radiation, like UV radiation, what UV radiation does is it causes, like, your A, T, C's, and G's in your DNA. So the non-ionizing radiation will cause T's that are next to each other to bind together instead of interacting with the A's across from them. So instead of the A opposite them, they will be creating covalent bonds next to them. So not hydrogen bonds across, but covalent bonds next to. So these are called thymine dimers. And that this is such an issue in the DNA that when you have your DNA strands being separated to make new strands and using each one as a template during DNA replication, that it causes problems with the ability of your cells to create copies of your DNA. And that this can lead to enough changes in the DNA and problems in the DNA that can lead to cancer. So thymine dimers are the issue with that. Your cells have mechanisms in place to repair that, but if damage is made at a certain point in the cell cycle and doesn't catch it in time, then that can lead to cancer, right? So this affects bacteria and stuff the same way, but they're only made up of one cell. So you can actually kill that organism if you bombard them with these types of radiation. This is just going over everything I just talked about as far as how these things work. So we can use ionizing radiation and UV radiation to sterilize. Obviously with ionizing radiation, that stronger radiation that causes our ions, that kicks off the electrons, that's really useful for sterilizing things that you need to penetrate. So now you can make your needles and package them in the little package and put the whole thing in an ionizing chamber of radiation to irradiate them. And then that ionizing radiation can get through the plastic, get through the needle, everything. So the whole thing is completely sterilized, all without having to treat it with the autoclave and risk melting the plastic or something like that. So you can actually have it sterilized in that package, and the whole thing is sterilized inside that package. So when you open it up, inside and out, completely sterile. So irradiation with ionizing radiation is actually really commonly used for our disposables. It's actually commonly used as well for food products. So food products will often be sterilized with ionizing radiation. So pretty common for that. Non-ionizing radiation, it doesn't penetrate. So UV radiation can't penetrate most surfaces. So if you're trying to sterilize with non-ionizing radiation, like UV radiation, you'll have to expose surfaces. So if you're working in one of those biosafety cabinets like we have in the lab, we don't really use that much. But if you're working in one of those cabinets, you'll turn a UV light on afterwards for like 30 minutes to an hour, and then that'll sterilize all the surfaces in there, but it won't penetrate, right? So it can be used for sterilization, it just cannot penetrate. So it's a little bit limited there. This is just a little picture diagram showing what the thymine dimer would look like. We can see we're not creating the pink bonds between the A and the T, we're creating between the two T's. It's actually a covalent bond that will form. Moving on to decontamination. If you filter something, you pass it through a filter, if your holes are small enough in your filter, you can sterilize things. So if it's small enough to keep getting viruses through, then potentially you can sterilize something by filtration. Filtration,
I didn't even think you could make something that small.
Yeah.
I guess you can.
yeah, you can. And so it's not cheap, right?
Right.
And so in order to sterilize liquids that way, it can be expensive. And depending on what product you're making, you might not need to sterilize with filtration, right? It might just be fine to just get most of the microbes out. But filtration can potentially sterilize. And it is a decontamination method because we're literally physically removing the microbes out of the liquids or the air, right? So you can filter air as well. So HEPA filters are air filters that work that way. Osmotic pressure, this is using those concepts of hypertonic environments to help draw liquid out of the cells to lead to plasmolysis. So the shrinking of the actual cell from losing the water is called plasmolysis, and this can kill bacteria. Obviously not a good way to sterilize because endospores are literally adapted to survive that, but it's a great way for preserving food and for decontamination. So you could do this with salt or you could do it with sugar, which is the whole point of preserves. You just have so much sugar in it. It has the same effect. Then we have our chemical control methods, okay? So that's it for our physical and mechanical. Now moving on to chemical or our chemical control methods. Most of these guys are being used in liquid form, but they can also be used in gas. Most of them, we don't usually use them in solid form unless it's like as a storage format for getting it, like solid soap. That's what I'm trying to get at here. So you might store soap as a solid, but when you're using it, the whole point is to kind of liquefy it, right? So whatever. But so we have two different kinds of solutions that you could have. If it's based in water, we call it aqueous. If it has any alcohol in it, we call it a tincture. So there's different levels of our chemicals. So we have different germicidal activity involved. So high level, and then we have intermediate, and then we have low. All right. So they basically go along with whether you are a critical item or whether you're semi-critical or whether you're non-critical. That's really what they go along with. And so if you are high level germicidal, these are things that kill all microbes, even endospores. And so these are used on our critical items. This would include catheters, heart-lung equipment, and implants. Whereas our intermediate level, the things that come, these are for things that might be our what we call semi-critical. They come into contact with the mucous membranes. This would include respiratory equipment or thermometers. And then our low-level germicides would be like our surfaces and things like that that come into contact with intact skin or otherwise healthy people. But you just want to eliminate the likelihood of contamination as a fomite, for example, right? So these will eliminate most vegetative bacteria and most fungal cells, but maybe not all of them. So we use these for electrodes, straps, furniture, doorknobs, things like that. Again, these are going to basically work on the same aspects of the cells that the other physical methods did. So again, attacking cell wall, cell membrane, nucleic acid, or proteins. Cell wall, breaking down the cell wall somehow, making the organism more susceptible to other treatments. Cell membrane, reducing that selective permeability. Nucleic acids, preventing you from making copies of your DNA or making your DNA to RNA, which is your transcription, or your RNA to your protein, which is your translation, or literally like denaturing existing proteins with heat or pH or something like that. So obviously when we start talking about adding pH, that's going to be on the chemical side of things, right? So we're going to start with our halogens. Our halogens are literally what we would consider the halogens on our periodic table of elements. Fluorine, bromine, chlorine, and iodine are the commonly used ones. We don't hear a lot about bromine that much, but if you have a hot tub, like they often use bromine instead of chlorine for hot tubs. Depends. I use chlorine on mine just because it's like just easier to buy. The chlorine is everywhere, but like I don't know. Which by the way, you can get like inflatable hot tubs. So my husband was like, I want to get a hot tub. And so I'm like, what if we don't use it? Like, how are we going to like whatever? So you can get like these inflatable hot tubs that are like actually pretty cheap. And we've had our inflatable hot tub for like 3 years and it's set up all the time and we leave it outside. So it's outside in like a covered area, but it's like all year round we run it. And so, I mean, depending on if you want to pay for the electricity bill on that, but like even when it's snowing outside, we can go out in our hot tub. It's just an inflatable little crappy old, 2 people can fit in it very comfortably, but you know, 4 people are just going to be a squeeze, but you're not going to get more than 4 people in there. Like it's pretty tiny. But if you have a house and you think you want to check out, if you think you'd be interested in getting a like full hot tub, those are quite pricey. I mean, you're talking like 10 grand for a really nice one. So they're not the little inflatable ones, man. Those are like a couple hundred. So one way to try it out if you're interested. We like ours a lot, and we'll eventually get an actual real hot tub, but just thought I'd spread that. We got off of Amazon. So yeah. All right, so halogens are fluorine, bromine, chlorine, and iodine. Obviously, fluoride in your toothpaste is an obvious one. Chlorine in pools. We also treat our drinking water with chlorine. We often pump a gas through it. It's like actual chlorine-based gas that we'll pump through our drinking water in order to treat that. So these are actually pretty effective. The halogens in general, if used at a high enough concentration and if used appropriately. So that's what a lot of the sterilization requires, right? You have to be following the guidelines if you want to sterilize with these. Otherwise, you're just disinfecting. But you can potentially sterilize with any of our halogens. So these guys can kill endospores if they're used appropriately. And this will include things like gas, chlorine, hypochlorites. If you ever heard of hypochlorous acid, it's a pretty common one that they use in skincare products now. It's made by your neutrophils in your cells. So it's a common biological acid that's used to kill microbes through oxidation and chlorine, basically. And so you can buy that for pretty affordably in skincare products. And it also has a very calming effect on the skin as well. So anyways, so these are chlorine-based compounds. These can potentially be used for sterilization at high enough concentrations. And these will basically disrupt enzyme function and denature proteins. Obviously, you have to be able to use them at certain concentrations in order for them to be effective. And if you're diluting them by liquid, like urine, blood, pus, and things like that, can dilute them and cause them to be less effective. So obviously, this would include things like bleach. And it smells like chlorine, right? So bleach is one of these things, and you can use bleach to disinfect. It's usually not at a high enough concentration in home use to sterilize, but it is very great, very, very highly recommended for disinfection. And if used appropriately and for long enough periods of time, could potentially sterilize. All right, iodine is one that we used to use pretty commonly on skin, but it's less and less used now because it can be irritating. So we had tinctures of iodine or aqueous solutions of iodine that would be used on skin surfaces. Basically, it disrupts the ability of the proteins to function, but it can be very irritating. Examples of these that are commonly used in medical environments would be betadine, povidone iodine, and isodyne. And these, again, if it's got that brownish color to it and it leaves that yellow tinge to the skin, that's iodine that's in there. People can be allergic to this, and this also can just be irritating in general. So they steer away from this now. It's not as commonly used, but we still see this used in like skin prep, like as an antiseptic in surgeries and things like that. Next, we have our phenol-based compounds. So phenol itself is also known as carbolic acid. It's what Joseph Lister used to prep his surgery suites and kind of was the father of using antiseptic technique in that regard. It's pretty good at inhibiting enzyme function and denaturing proteins and things like that, but it's incredibly toxic and it can be incredibly irritating as well. Phenol, man, I love the smell. It's what that Carbofusion smelled like, that chloroseptic throat spray, like that sort of smell. I love the way it smells. I think it's just like a memory, like, a nostalgia thing. But yeah, so that's phenol, but again, can be very irritating at the concentrations needed to actually be disinfectant. So we do use this in some rare cases. We might clean out like pet cage situations and large like facility type cleaning with phenol, but we don't see it a lot used in like smaller applications, and we certainly don't use it as an antiseptic. But phenolics themselves, which are molecules that are derived from phenol, can be used as antiseptics, but it can still become irritating. So chlorhexidine is a great example of this. Chlorhexidine is, if you ever donated blood, if you donate blood, they like take a swab and they rub on your arm for like 30 seconds, for a whole 30 seconds. And they're supposed to be doing it in a 2x2 square. And they use chlorhexidine in that preparation. And so that's the idea, is to get you as, decontaminated as possible, the antiseptic there. So we're using antiseptic as well as physical removal of the microbes, because they'll go from the center and they'll go outwards and then they'll continue to rub around. So we have both actions there, the actual chemical action as well as the physical action. If you're allergic to chlorhexidine, which is possible, then if you donate blood, they will use iodine. If you're allergic to both, then you can't donate blood. There's just nothing that they can do. They're putting out 16 gauge needle is I want to say a generous term for what they use. It's more like 14. So the smaller the number, the larger the needle. So the bore on the inside is 16 gauge, and it has to be so that we're not shearing the red blood cells. Because the whole point is to collect your red blood cells and not break them down. So the actual outside measurement of the needle is closer to 14 gauge. To put it in perspective, my nose ring, like most septum piercings or body piercings, are typically 16 gauge. So it's smaller even than the needles that you use when you donate blood. And that's to prevent the wrecking of those blood cells. So when you guys go into nursing school and you think that you're sucking at phlebotomy, go talk to somebody that draws blood for like OBI or something, because they're using massive needles, 30 sticks a day, man. Like they know what they're doing. So if you need tips on how to draw blood, and now you could say all day long that you want to, that, well, they're drawing blood on healthy people. Well, I was a phlebotomist at for DLO at Integra Southwest. Those are not healthy people. And I can tell you, my experience at OBI dramatically improved my ability as a phlebotomist. I was an excellent phlebotomist. And it's because of my experience at OBI and having to stick with those massive needles, and they have to stay in there and draw blood for a full 5 to 10 minutes, not just for like the 2 minutes that it takes to fill up your tubes. So just be aware of that when you guys start getting calls from the labs at the hospitals that you work at, whether you're a vet or you're at a hospital, it doesn't really matter. But if you're hemolyzing your blood samples, the lab will call you down and have you redraw your sample if it's overly hemolyzed. What that means is you've broken down those red blood cells. I cannot tell you how many times I've worked in a hospital lab where I will have to call the floor and be like, I need the samples redrawn for John Doe. And the nurse will be like, well, how did you hemolyze my sample? Nope. Nope. Impossible for the lab to hemolyze. I'm just saying, we don't put it through any procedures that could possibly hemolyze it. There's nothing that we do to it that could. It's the nurse drawing the blood that hemolyzed it. So you would do that by sticking that person multiple times or causing damage to the tissue when you're sticking. If you're taking a butterfly, that's a 22 gauge needle. It's very tiny and can shear the blood cells just on their own. And if you're not putting it into the vein correctly, the position of the needle or the damage that you cause to the tissue can cause the blood cells to be damaged, and you'll have hemolysis from improper phlebotomy technique. That's what hemolysis is from improper phlebotomy technique. So if you guys suck at phlebotomy, talk to a phlebotomist, because that's what they do. If you're lucky enough to work at Mercy, they have floor phlebotomists. If you work at Integra, someplace like that, then nurses often do their own blood draws. If you're on the floor, if you're in the ER, they have like nurses or nurse aides that like are better at phlebotomy, but they'll have go draw the tubes in the ER before the nurse comes in for the typical nurse practice stuff. But that's on the ER. If you're working on the floor, you're going to be drawing your own tubes. So get better at phlebotomy. So if you're sucking at that and you think it's okay to suck at that, I'm telling you as a patient, it is not okay to suck at phlebotomy. It's not, okay? So work on your phlebotomy skills. So anyways, moving on, chlorhexidine is derived from phenol. It's not phenol, but it has the same chemical derivatives. And we find chlorhexidine, of course, it has chlorine in it as well, so it has that other aspect going for it. But it's based on the phenol chemical structure. Hibiclens, Hibitane, and Peridex. These are all basically commonly used forms of liquid based on chlorhexidine. And so Hibiclens is like a soap that we use for cleansing skin that could have staph. So if you have a staph infection, we'll often instruct our patients to wash themselves with Hibiclens. It's also the soap that we use in surgical scrub. So if you are a surgeon or if you're working as a scrub nurse or something like that in a surgical environment, you'll be scrubbing with chlorhexidine prep. So it's Hibiclens typically that they're using for that. And the scrubbing action itself helps remove the microbes. So that's the physical side. The chemical side is the chlorhexidine in it. Okay. Then we have also the sudsing of the detergent. Like Peridex, you can use mouthwashes with chlorhexidine in it that are very, very highly recommended by dentists to help with the oral hygiene side of things. It's low toxicity, but you don't want to ingest this. Okay. Alcohols. So alcohols are effective at breaking down the cell wall and the cell membrane and degrading and, well, I don't want to say degrading, but coagulating proteins. So causing proteins kind of lose their normal structure and then sticking together. So they can do all three of those things, but it's still not sterilizing. You don't sterilize with alcohols. So obvious alcohols, we know isopropyl alcohol and ethyl alcohol, so ethanol. And those are two very commonly used ones in microbial control. If you have 50% or higher alcohol concentration is when you get to that lipid effect and the cell wall effect. But you don't want to go 100. Absolute 100% alcohol, it'll dehydrate the cells and it'll inhibit growth, but it is not recommended for antisepsis. We recommend 70% alcohol. And the reason why is because that water that's in the alcohol helps with the coagulation of the proteins, just like the moisture in the hot moist did compared to the dry. All right. So you want the water there to help with the coagulation of the proteins. So when you guys are out there buying your antiseptics for home use, 70%. Okay. It works better. 70%. You use hand sanitizer. So typically, most commonly, isopropyl alcohol works better than ethyl alcohol, and it's cheaper. Okay, so why are most of, like this one here, just says alcohol. So great, that's not helpful. But most of them that we look at nowadays that you buy will say ethyl alcohol in fact. So why do we use ethyl alcohol now when we used to use isopropyl alcohol? Anybody else notice that? Hand sanitizer used to be isopropyl alcohol, and now it's ethyl alcohol. So the reason for that is that ethyl alcohol is more biologically friendly in the sense that it's not going to dry out your skin and ruin your skin barrier as much. So ethyl alcohol is more friendly to your skin barrier. Use isopropyl alcohol, it's more effective against the microbes, but it's also more effective at drying your skin out. And so with the COVID era, people were using hand sanitizer so much that they didn't want people using isopropyl alcohol because it would dry out their skin and make you more susceptible to other infections, and especially in the healthcare industry, right? So that's why we switched to ethyl. Even though isopropyl is more effective, we're using it so commonly now that they recommend ethyl. All right. Oxidizing agents are things that rely on oxidative free radicals based on oxygen, literally. So we learned about a little bit about this when we talked about aerobic microbes versus anaerobic microbes. But if you're an aerobe, you need to have things in place to deal with toxic oxygen byproducts. And this includes hydrogen peroxide. You need to be able to protect from that. You need to be able to protect from any of the oxygen free radicals. And we can actually use these as methods to control microbes. And we know hydrogen peroxide can be an effective way to be an antiseptic and things like that. So we can actually have these be sterilizing in high enough concentrations. As it turns out, the typical use of hydrogen peroxide at 3% is not enough to sterilize, but it's very good for disinfecting, especially for taking care of anaerobes. And it's highly recommended by your dentist to use a hydrogen peroxide-based mouthwash. Really effective for controlling the microbes in your oral biome. So it's great for treating anaerobic bacteria. If you can get up to 35%, I don't know why it's written out as 35 and not just the number 35, but whatever. 35% concentration of hydrogen peroxide can be used for sterilizing. And we can actually vaporize it and turn it into we could fill this whole room with hydrogen peroxide vapor, 35% hydrogen peroxide, and sterilize everything in the room all at once. And a situation like that would be called a chemiclave. It's like the autoclave, but with chemicals, chemiclave. So hydrogen peroxide, again, it makes our toxic free radicals that will basically assault other molecules in the body and steal electrons and things like that and interact with those molecules. So it can be at high enough concentrations sterilizing, but otherwise it's disinfectant. What else do I say about this? We're pretty familiar with hydrogen peroxide. We'll come back to our detergents. This will include things like quaternary ammonia compounds. So our detergents, this includes surfactants. So we have detergents essentially working with those polar heads and the non-polar tails, just like our phospholipids. Surfactants qualify as a detergent. So there's other detergents that have a sudsing factor that are not the polar head and the long tail, or they might operate differently than typical surfactants. But for our purposes in this course, they're the same thing, okay? For chemical, if you want to go into chemical engineering, go discuss the differences with somebody else. But for our purposes, surfactants and detergents are interchangeable, okay? So these are our sudsing guys. And our sudsing guys, basically their structure of their polar head and their long non-polar tail, the hydrophilic head and hydrophobic tail, look a lot like our phospholipids and can kind of stick in between our phospholipids and cause the disruption of the membrane, which we showed earlier, right? And that's the idea. So these are chemical guys that actually interfere with the structure of the cell membranes. A lot of these guys have an actual positive head So they're more likely even to interact with the negative heads. So the negative heads will hang together because they're charged, but they're negative and to negative. But when you introduce that positive head, that really wants to get in there and break in and disrupt the chamber. So they have a positive head from this nitrogen thing. So the nitrogen can bind onto 4 things. And when nitrogen binds onto 4 things, it's called an ammonia compound. These are all, that's what a quaternary ammonia means. It means to have N with 4 things, quaternary, 4. And that's what gives it the positive charge. And then the tail is hydrophobic. That's a quaternary ammonia compound. A quaternary ammonia compound is a detergent or surfactant. All right, and so they're going to break down the membrane. These are very weak microbicides. They're not very useful for killing the microbes, but they're great for weakening the cell membrane so that other chemicals can get in there and have the effect on them. All right, so that's the basics of it. But there are microbes that actually can like grow in the presence of these soaps, like Pseudomonas, which is a common environmental. It's everywhere. It can grow on soap dishes and then like the soap on your. That's why it's soap dishes can sometimes get nasty. It's usually Pseudomonas. Heavy metals can bind in the active sites of our proteins and act as competitive inhibition. This guy will include mercury, silver, and gold, but obviously more commonly we're going to see mercury and silver. It's actually pretty common to see mercury in a lot of like ophthalmic solutions, like for contact lenses and things like that as a preservative. I want to be very clear that mercury is all over in our environment and all sorts of things that you eat and drink and all sorts of things. And I also want to be very clear about something else. The mercury that was ever used to make vaccines is at an abysmally smaller amount than what you will be exposed to on a daily basis just from eating and drinking. So if your body can't handle a vaccine that has that much mercury in it, believe me when I tell you, you've got other problems because you won't be making it in the world. So the next time that somebody tells you about mercury in a vaccine causing autism, ask them, okay, well, are you eating food and drinking beverages? Because you're probably getting 100 times as much mercury on a daily basis than from one vaccine. Just saying, so that we're educated about this. That's not an argument against vaccines. And there's actually absolutely zero evidence that it's even correlated with vaccines. So people come to me and they're like, well, there's that paper published. Yeah, you mean the one that was retracted by the guy that no longer can practice medicine in the country he's from? That guy? Is that who we're talking about? The same? Because last time I checked, that's a load of shit. And I don't know why people are holding on to it for their dear lives. Yeah. Because people have autism and they want an explanation for why. And there was that one lie that was literally paid. He was paid to publish that lie. He falsified all of the evidence. And we've had paper after paper after paper after paper after paper, peer reviewed from many different countries from all over the world that have repeatedly shown there is absolutely no correlation. Let it go.
And didn't Scott lose his medical license?
Yeah, he lost his medical license, but he can still post, videos on YouTube.
Well, yeah. I would love to see your responses on that stuff that's been posted on Facebook lately about vaccines.
Wouldn't you though? Wouldn't you? So we'll be going over that stuff. I don't want to get too deep into it because we're going to be talking about vaccines in class, and we will be talking about that stuff. It is my job to make sure that you as healthcare workers are educated on how vaccines actually work. It's my job to make sure that you are properly educated on that so that when you go talk to your loved ones or your family members who are anti-vaxxers, or you're trying to talk about why you don't agree with RFK's approach to removing vaccines from America. And if you haven't been watching, this sounds like I'm making something up. I promise. Yeah, if you're not watching the news, y'all, you need to watch the news because this is going to affect you directly. Because we have RFK in office, and I am not arguing against the whole administration. You can have your feelings about the administration, whatever you like. But this particular policy about vaccines is disgusting because we have an anti-vaxxer that is in control of healthcare right now. And he has been a well-known anti-vaxxer since well before he was appointed. And he is trying to skirt around the anti-vax issue, but he's already canceled many meetings about making sure that Americans get vaccinated and get their vaccination sequence, including we will not be having, we have already canceled several meetings on flu vaccines for next year. So we probably won't be having flu vaccines for next year. It's been canceled. We've lost funding for the CDC. The CDC has been told to stop, giving us information about outbreaks around the world. educated. Okay, that's all I'm gonna do. I'm gonna say that and leave it at that. We're almost done. Aldehydes. Aldehydes, because I promise you it can go on forever. And I promise you, if you are anti-vax or anybody you know is anti-vax, come talk to me and I guarantee you I will knock down every point you have to make. Guarantee you, yours are opinion-based and mine are fact-based. Guaranteed. This is coming from somebody, of course, you could say all day long, well, yeah, you studied under the guy that developed the COVID vaccine. Yes, you're right, I did. So I know my shit, bet you don't. That's what I have to say about it, all right? So aldehydes, these are chemicals that can degrade proteins and disrupt the proteins. Sorry, I have the soapbox and I can't come down from it. So glutaraldehyde and formaldehyde are a potential for sterilization. Glutaraldehyde and formaldehyde can sterilize. And we can use glutaraldehyde a little bit more broadly for cleaning surfaces and objects and things like that. So it can sterilize things that would otherwise be sensitive to heat. But formaldehyde is incredibly damaging to tissues, living tissues. So we can use formaldehyde and formalin, which is the aqueous version. So, dissolved in water, thank you. We can use that for an embalming fluid for dead bodies, but you would never use formaldehyde or formalin on living tissues or around living tissues. Orthophthalealdehyde, it took me forever to even be able to say that word, and you are welcome that I'm not making you spell it out. OPA, or orthophthalealdehyde, this is similar to glutaraldehyde. It does not sterilize, it's less irritating, and it's more expensive. It doesn't sterilize, but if you're using it on environments around living organisms, it's a little bit more friendly for living organisms. All right, then we have our gases. This includes ethylene oxide and a chlorine dioxide gas. So these are chemical gases that we can use to fill entire rooms and then sterilize everything in the room. So these are useful for actual sterilization and can kill endospores. Ethylene oxide is incredibly effective at sterilizing, actually. You can fill entire building with ethylene oxide and then sterilize that building. However, it is explosive and it is carcinogenic. So it's hard to work with it. Yeah. So
should we?
What?
Should we do that though? We could
What do you mean?
fill the whole room with it.
So this is for sterilizing like large equipment and things like that.
Oh, I guess, yeah.
Huh? So they don't use this very commonly. They might occasionally cycle operating rooms with treatment with these types of gases. It's less common, but what you see this used very commonly in is BSL-4 labs. So those labs, like at the CDC, when they're working with like Ebola and things like that, where they work with the suits, they work in a room that is, it's like a positive pressure room. So like if you got a tear in your suit, that I guess is negative pressure, the room is negative pressure. So the air would rush out of your suit. You constantly have air blowing through your suit. So if you got a tear in it, air would rush out of the suit so the air couldn't get in. And then the whole room itself is negative pressure to like the outside environment. And also if there was a rupture in the room, air would rush into the room and not out. So it's controlled with air that way. But they cycle from lab to lab to prevent too much contamination within each lab. So like every few weeks or something, they'll cycle to a different lab, and they have 4 labs that are BSL-4 positive, and they'll only operate in one at a time. And when they leave that lab, they sterilize the whole lab with ethylene oxide, so that the whole lab is completely sterilized, and they'll expose it to that for like a whole day. And then they have to vent it out and they have to treat it on its way out so that it's not contaminating the environment. But that's a more common situation where we have something like that. Or for example, with chlorine dioxide, which is not explosive or carcinogenic, but it does sterilize as a gas, they use that to sterilize the Senate offices after somebody mailed anthrax to the Senate. And so it's endospores. They have to get rid of the endospores. You have to sterilize. All right, acids and alkalis, we know how this works. This is pH alteration, so disrupting those attractions that are associated with hydrogen bonds. So we know this is commonly used in foods. Anytime that you look on the back of the package and you see sorbic acid or propionic acid or lactic acid or acetic acid or ascorbic acid, which is vitamin C, or citric acid, which is citric acid, these are all added as preservatives because that pH can help with throwing off the microbial enzyme functions. And because of that pH destabilization, disrupts the hydrogen bonds. So acetic acid, if you didn't know, that's vinegar. That's what makes vinegar an acid. So this is why we recommend you cleaning your house with vinegar, because it's very safe, but it's still very effective at disinfecting. But our acids and alkalis typically won't sterilize, not at the levels that we would work with them as people on average. I mean, if you're using concentrated sulfuric acid, yeah, it's going to sterilize, but it's also going to eat through your table and the floor and like all that stuff. So yeah. Here's our little summary of some commonly used things.